What's up, everyone? I wanted to give you all some advice on the Biochem section of the MCAT. I know it's a very hard section of the MCAT, and some people might be confused as to why they're performing so poorly on that section, even though they're bio majors. And one of the things, having scored a 96th percentile on that section, and the score is right here, one of the things that I could say about Biochem is that a lot of your success on that section revolves around how well you can interpret figures and passages. Oftentimes, I would highly recommend reading a lot of scientific literature in order to get really good at this section, because a lot of the figure interpretation you will use in those hard science pieces of literature will help on this exam. My goal is to go over a passage that's going to help you all understand what I mean by that, because Biochem is really all about having a good amount of content review, but then it's also about passage interpretation. And I would argue that some of the hardest passages on the entire MCAT are found in Biochem, because the examiners know that most people taking the MCAT exam are somewhat of a bio major and they have somewhat of a familiarity with biology. So they're going to put more an emphasis on interpreting harder figures rather than just asking what is the function of the mitochondria. So that's really what Biochem boils down to, as really the pieces of advice I have to give towards it. Yes, there are some calculations that are involved in Biochem, but they're far and few in between, not nearly as much as ChemPhys, which is why more of my advice would revolve around the fact that you should know your content very well, especially content in relation to difficult areas like the kidneys, the hormones, as well as other areas related to like microbiology and immunology that may not pop up too much. And embryology was a thing that I personally struggled with and still struggle with, to be honest. Developmental biology is something that can be tested, especially what the endoderm, ectoderm, and mesoderm, which tissues derive from those. And to be honest, I barely remember it, even after taking the test and doing fairly well on it. But my advice when it comes to Biochem is mainly to get good at interpreting the figures and ask yourself why you're getting questions wrong and review them thoroughly. So without further ado, we're going to hop right into a passage so you can see how someone like me, who did very well in Biochem, is able to break it down. All right, so we're on the Jack Westin passage right here. And I'm going to go under where it says sections and pick Biochem. Biological, biochemical. That's what Biochem is for AMC passages. And why don't we do one about the Ebola virus? I hope we get a figure out of this one. We'll see. Sweet. Yes, we do. 4 questions and we get a figure. So without further ado, just going to scoop my computer up and I'm going to break down this passage as many of you would like to see. Ebola virus infection is initiated by the viral membrane glycoprotein GP mediated fusion of viral and host cell membranes, allowing for the injection of viral genomic RNA into the infected cell. Mature GP is a trimer composed of 3 GP1, GP2 disulfide linked heterodimers. Telling us the composition of it. GP is considered mature once the GPO, uncleaved GP precursor peptide, is hydrolytically cleaved into GP1 and GP2, allowing further combination into GP. GP1 specifically mediates adhesion to host cells and is proposed to regulate the transmembrane subunit GP2, which is thought to carry out membrane fusion. Okay, so they're telling us the function of essentially this glycoprotein and how it's very important when it comes to the Ebola virus infecting and how it matures too. It's a very general idea. I don't have the specifics memorized in my head, which is why we will go back to the passage as needed to solve these questions. Researchers proposed the mechanism by which GP2 mediates membrane fusion relies on the capsin, I don't know how you say that, family of acid-dependent proteases. So they're talking about the mechanism. It relies on some sort of family of proteases. That's what I'm getting. Specifically, cathepsin B. Oh my, how do you say this? I'm just going to call it Cat B for short, because a lot of times in the MCAT, you will get words that you don't know how to pronounce very well. So I'm going to call it Cat B. specifically CAT-B, and there it has the abbreviation, and CAT-L. So these are two, what are they again, just to reiterate. They propose a mechanism by which GP2 mediates membrane fusion relies upon the CAT family of acid-dependent proteases. So we're talking about specific proteases here called CAT-B, CAT-L. CAT-B and CAT-L can be activated in the endosome before localization to various parts of the cell. These proteases are thought to facilitate cleavage of a highly variable C-terminal region. I'm going to highlight cleavage. C-terminal region. After the removal of mucin-like domains from GP. To examine the role of these enzymes, it's very important. Researchers tested the infectivity of vesicular stomatis virus particles containing Ebola virus particles lacking, they're doing a knockout, the critical like mucosin-like domain. They put Ebola V, and then in Biochem, whenever you see this delta, it just means they deleted it. So what they're trying to say is that they have the GP, but they lack the mucin-like domain, as they indicated right here. And their own host-binding glycoprotein, which fused to the membrane independently of the Ebola virus GP protein. On the varicose line, in the presence of protease inhibitors, okay, I'm going to highlight protease inhibitors, CA074 and FYDMK, and then they say it's in figure 1. The effects of the inhibitors on CAT-L and CAT-B in varicose cells are shown in figure 2. Okay, so we gotta slow down and kind of take a step back because they threw a lot of information at us. So they're talking about in figure 1, relative infectivity of VSVG, which was what? The host binding glycoprotein, and EBO GP delta muc. So these are particles that lack the mucin-like domain. Okay, so it says relative of VSVG, and then lacking the mutant, and then strains after the addition of the inhibitors of the proteases. So here's what they're saying up top. They're talking about the relative infectivity. So they're saying that the host binding glycoprotein, that is what VSVG is, the host binding glycoprotein, it has normal infectivity from the looks of it in both cases. Whereas it's saying that the Ebola virus particles lacking the mucin-like domain, their infectivity drops quite a lot, which is quite interesting to note right there. I'm just trying to go back and see how CAT-B, CAT-L are related to them. It says that they are facilitating the cleavage of a highly variable C-terminal domain after the removal of mucin-like proteins. So it seems like that these guys, they require mucin to be removed because it says that they are involved in cleaving those C-terminal domains after the removal of mucin-like proteins from GP. Yeah. And then it says they tested the infectivity by containing Ebola virus lacking the mucin-like domain. So that would mean that if they lack the mucin-like domain, they would likely start binding to the, if they lack the mucin-like domain, chances are that means that the proteins will start likely being cleaved a bit earlier compared to the host binding glycoprotein. Okay, this is starting to make a bit more sense. Whereas, let's see, it says CAT-B and CAT-L enzyme activity in varicose cells after the addition of CA074 or FYDMK. That's what they're talking about, the activities right there. So what are they talking about? CA74. So they're talking about the activities of CAT-B and CAT-L. So we see that CAT-L doesn't really notably decrease too much, at least the activity, when you add more of this, the CA inhibitor, but CAT-B does. Whereas vice versa, with the FYDMK, it seems like that this inhibitor is more potent with CAT-L, but not CAT-B. So actually, it does switch. It switches between the two of them. You have to watch out what the circles represent here, with what's going on. Because Cat B decreases in the first inhibitor and then not as much in the second, but Cat L doesn't really decrease in the first inhibitor, and it does notably so in the second. Okay, I think we have a good conclusion of what these two figures mean. So what we can do is start going on to the question stems and attacking them and seeing how the passage will relate in that regard. So without further ado, which of the following is true regarding the proteases used in the study? So they talk about the proteases Cat B, Cat L. That's essentially what's going on. And I see in the question stems that they're talking about the inhibitors, the FY inhibitor and the CA inhibitor. I'm just going to call them that for short. So all of the answer choices are talking about that. So chances are we might only need to pay attention to figure 2, given that all the answer choices are talking about the inhibitors in common. I'm not selecting or committing to an answer choice yet. I'm just seeing commonalities in them that allow us to narrow down the information we need to get in the passage. So let's see. Cat B is selectively inhibited by FY. Is that true? Let's see. I don't know, because it actually says Cat B is inhibited by CA, so I'm not really liking it. And then Cat L selectively inhibited by CA. They actually switch the relationship around, which is why I don't like A that much. Yeah. And then Cat B is selectively inhibited by CA. That is true, it does go down. While Cat L is selectively inhibited by FY, the FY guy. I see that Cat L, it does go down. I do like it a lot. But then I also do see that Cat B is inhibited as well, because technically, I'm going to leave B alone and see what the other answer choices say. Because technically, the FY inhibits both, but it doesn't inhibit Cat B as greatly as it does Cat L. B could be the right choice. We'll just have to see. Cat B and Cat L are both non-selectively inhibited by both inhibitors, which I don't think it's entirely true, because for the first one, we see that Cat L levels don't really decrease all that much. Whereas in the second one, I could kind of see it being a little more true, because the Cat B levels do have a marked decrease. So I think it's incorrect to say that they're both non-selectively inhibited, because at least the CA inhibitor selectively inhibits Cat L. D. Cat B is non-selectively inhibited by CA 074 and FY DMK. By CA and FY, so they're saying Cat B goes down, which is true. It does decrease in both just about the same. And I guess if we pay attention to the y-axis, it actually takes less of the FY inhibitor for the activity to go down compared to the CA, which could lend stronger support to it being D more than B, because the FY inhibitor, it takes less of it to inhibit both enzymes. And anyway, it says Cat L is selectively inhibited by the FY, which I agree, because Cat L doesn't go down under the CA. Because we do have to pay attention to the y-axis, and the FY inhibitor does decrease both enzymes a lot sooner than the CA decreases the activity of Cat B, I think D is going to be a good choice, if that makes sense. I'm getting the sense that FY is just a stronger inhibitor for both of them. Yes, even Cat B, because the activity is around 800 or a little below 800, only with 1 micromolar of the FY inhibitor. Whereas with the other inhibitor, the activity reaches the same point at around 10 times the concentration, meaning that FY is a more potent inhibitor. Wow, that was a tough one. Many people selected B, and I don't blame them, But you have to watch out as to the trends of the graph and see where the decrease is happening, because Cat B did markedly decrease below 400, roughly a little less than 1200 to a little less than about 200. So the activity, it was definitely a fraction of what existed before, which is why it's a non-selective inhibitor. That was a tough question. Don't blame y'all if you got it wrong. But this is why we have to really think and see the data for what it really is. The final step of GP maturation depends on which of the following processes. Well, that's very nice because we looked at what GP maturation requires. They said it is considered mature once GPO, uncleaved GP, precursor peptide, is hydrolytically cleaved into GP1 and GP2, allowing for their combination into GP. So my thought process is that between all of these answer choices, glycosylation doesn't seem like a good choice because it doesn't ever talk about that we just add like sugars to it. Same with alternative splicing. We're dealing with proteins here, not RNA post-transcriptional modification. Not a fan of B. Proteolysis, however, does seem like a really good choice because they literally said that the peptide is hydrolytically cleaved. So I like C a lot. D is not a good choice. Reduction by NADH, we're not dealing with metabolism here, which is where we would see that happening. Proteolysis is the choice. Sometimes in biochem, you only need a small portion of the passage, a couple words as a matter of fact, in order to get the question right. Because all you need is precursor peptide is hydrolytically cleaved. That is all the evidence you need to get the question right. Once I started realizing this, I started scoring way better on my practice tests and on the real exam. Moving on to question 3. Mature GP would produce how many bands on SDS page after treatment with BME? Yeah, let's go and see what BME stood for. Wait, I think we need to know BME off the top of our heads because BME stands for beta mercaptoethanol. And so what that reagent is, beta mercaptoethanol, is that it's going to be an agent that breaks disulfide bonds. So the question to rearrange it is asking, how many subunits does mature GP end up having such that we could cleave the disulfide bonds and it's going to create 2 different fragments or more, again, multiple fragments on the SDS page. I don't want to make assumptions yet about the answer choice. So we would probably need to read the answer choice, this last part of this paragraph, the first paragraph again, because they talk about the maturation, because the question is asking about mature GP. So they said that mature GP is composed of 3 GP1, GP2 disulfide linked heterodimers. All right, I think that might give our choice. That is what they said. It's considered mature once GPO is cleaved and is hydrolytically cleaved from GP1, GP2, allowing for their combination into GP. And then they talk about GP1 and then the function of GP2. But mature GP, they're saying it's a trimer. So it has 3 subunits composed of 3 GP1, GP2 disulfide linked heterodimers. Okay. I want you to pay attention to the part where it says it has GP1 linked with GP2 via the disulfide bond, because the answer lies in what's linked by the disulfide bond. Because here's the thing. We have 3 subunits. Each of the subunits is made up of 1 GP1, GP2 dimer. So if we were to cleave the bonds, we would all of a sudden have 3 GP1 proteins and 3 GP2 proteins. So 3 GP1s would go here, 3 GP2s would go here. What happens when we run them on an SDS page is that the 3 GP1 proteins would all produce 1 band, assuming that they are of a different molecular weight than the GP2. GP2 would produce a different band, assuming that they're a different molecular weight, which, let's see if the passage mentions anything. I'm not really seeing anything on the passage, but that's not too unreasonable of an assumption right there. So the answer would end up being 2 subunits, 2 bands, because all of the three GP1 subunits would form together to make one band. They would overlap, whereas the three GP2 subunits would overlap to make a second band, assuming that the protein is either lighter or heavier than GP1. We have to make the assumption that they're a different molecular weight. The passage never really said that. It did say that they're a dimer, but it never said that they were the same molecular weight. So because of that, I'm going to pick 2 for that reason. Wow. C was a tricky one, not gonna lie. Because reason why I think people picked it was because they said, oh yeah, it's a trimer, let's pick 3. That's not always the case, because you have to do a little bit closer reading into what the trimer is made up of. 3, GP1, GP2, and then the key is where the disulfide bond is. The disulfide bond is between the GP1 and GP2 proteins. So you're breaking apart the disulfide bond and making these proteins into 2 different fragments. That is why the answer choice is 2. 3 was a very tricky question. If you get this question right, the exam will reward you very heavily for it. Wow, this is a very good explanation. Yeah, beta-mercaptoethanol, BME. That's a very tricky thing to know as well in the passage. Sometimes they don't even write out the full name. Luckily, I was able to determine it, and this is where content should come in to help you out. You need to know what these reagents do if you're quizzed on them on the exam. All right, question 4. Which of the following observations would be a suitable control to confirm the activity of CAT-B, CAT-L inhibitors in vira cells infected with VSVG? Question 4 is one of those questions that's trying to make us think quite a lot, because it's what I like to call an experimental question. It's asking us, how can we make a good scientific experiment? And these questions can be a bit trickier. So they're asking which of the following would be a suitable control, and we want to confirm the activity of the CAT-B and the CAT-L inhibitors in the viral cells infected with VSVG. So we know that VSVG, it doesn't really get all that infected initially, at least, actually, sorry, it does. The rate of infectivity goes up quite a lot when we have it. I think we might have to use figure 1 as well, because what they're trying to say is that CAT-B and CAT-L, they function in the cleavage of the C-terminal domain after the mucin domains have been removed. That's essentially their function. Yeah, these proteases. Which makes sense, because when the mucin domain is removed, the Ebola virus, its infectivity goes down a lot. That's what figure 1's trying to say. So we're trying to confirm the activity of CAT-B, CAT-L. Now the inhibitors, their function is to stop the cleavage of CAT-B, CAT-L, which is why the enzyme activity decreases. So here's what I'm trying to think, because they're saying which of the following would be a suitable control. Let me think about what a control can do, because a lot of times in negative controls, they're meant to take away the independent variable to confirm that there's no outside or confounding factors influencing the experiment. That's essentially what's going on right there. Yeah, I just need to read up on what CAT-B, CAT-L are. It says that they are the dependent proteases. That's really what they are. All right. And then GP1 is part of the viral glycoprotein. Yep. They can be, CAT-B, CAT-L can be activated at the endosome before localization in various parts of the cell. So from the looks of it, it doesn't really say all that much, but CAT-B and CAT-L seem to be involved in at least they seem to be involved in degrading the virus. So chances are that they're actually produced from the cell. That's essentially what's going on. The reason why I say that is because that's making me not too keen on A and B, where we're talking about mature GP fragments, the levels of the cleaved fragments, because we're trying to see if cat B and cat L, they are a type of cytosolic protein fragments. That's what I'm trying to get at. So I don't think A and B would be a good choice because we're not trying to compare cat B and cat L, the inhibitors, to something that is completely unrelated to it, which is the GP fragments. Yes, cat B and cat L, actually, cat B and cat L aren't really even involved in cleaving the GP fragments. They're just involved in, they're involved in cleaving the virus. That's what they say, but they're not involved in the maturation of it. So I don't like A and B that well. Yeah, because they say it's considered mature if the peptide is hydrolytically cleaved. But cat B and cat L were never said to play a part in the maturity of the protein. Yeah, it just says it's hydrolytically cleaved by something else. We don't really know what. So because of that, we can't make an assumption A and B are right. So yeah, they're trying to say what would be a suitable control to confirm the activity of the inhibitors. Well, here's the thing. In the experimental control, we already have the presence of cat B and cat L. That's the thing. Which is making me trying to think about the difference between increasing the protein fragments, which is, which indicates that it's cat B, the remnants of cat B and cat L, at least what they have cleaved. Because that's what protein fragments are. They're leftovers of what the protein has cleaved. Or it says decreased level of the protein fragments. Here's what I'm thinking. If you increase the level of cytosolic protein fragments, that would mean that cat B and cat L, that they are still undergoing their function as proteases. But if you have decreased level of cytosolic protein fragments, well, that could possibly mean that the inhibitor might be it could be effective, which is confirming the activity of the inhibitor. Because they're talking about which of the following observations. It's asking which is confirming that the inhibitors actually work. Because the function of cat B and cat L is to cleave proteins. They're proteases. Yeah, they're not involved in the maturation of GP. They're rather involved in cleaving at least a certain part of Ebola, of the virus based on the passage. So confirming that the inhibitor works, you would actually expect to see a decreased level of the cytosolic protein fragments because it's trying to say that it's inhibiting the protease from performing its function. And what do proteases do? They cleave fragments. C would be if the inhibitors did not work properly. Yeah, which I'm not a fan of. Sometimes these questions, they have crazy mental gymnastics. Even for me, I had to think about this one quite a bit. But this is why you have to interpret the passage and hopefully it makes sense. Let's see if I got this right. Wow. Only 25% of students got this. Chances are, if this were to appear on a real MCAT exam, it would only be an experimental type question. Because this is a very low amount of students getting the question right. However, here's the thing. You don't know which questions on the exam would be counted as experimental. So you really have to solve it as if it were like any other question. Bottom line is, get good at answering very tricky questions that are related to experiments. Because, oh man, I personally struggled on those quite a lot during my studying days. And if you can learn how to improve on those types of questions in biochem, you will start scoring in the upper echelons, up the upper percentages of percentile scores on the exam. The examiners know that you're probably a bio major, and if not, you're something related when applying to medical school. So they make it a bit harder for regular bio majors to succeed on biochem. Don't be discouraged if you're a bio major and you're not doing that great on biochem. You just need to learn how the test works. And that right there is my passage review. And I think that's a wrap for this video. Please be sure to watch my next video on Psych Soch, because that is the next passage I'm going to break down. In the meantime, happy studying, take care, and I'll see you all in the next one.